Hello, my name is Dalton Jenkins. I'm going to be giving a lecture that I've prepared on chapter 32 of our textbook, which is titled An Overview of Animal Diversity. So let's talk about some characteristics of animals. Like, what makes an animal an animal? Well, animals are heterotrophs, but they feed differently than fungi. So we saw in previous chapter that fungi are heterotrophs that secrete enzymes and break down their food sources of organic materials outside of their bodies before absorbing their nutrients. Animals, however, ingest their food sources and they break them down within their bodies before absorbing their nutrients. Animals are multicellular. They're eukaryotes. Their cells have nuclei, but their cells do not have cell walls like the cells of plants or fungi. Most animals reproduce sexually, so a sperm cell from a male parent fertilizes an egg cell from a female parent, and this process forms a zygote. The zygote will then develop into a blastula through a series of cell divisions known as cleavage. The blastula will then develop into a gastrula through a process known as gastrulation. And in gastrulation, the embryo becomes layered, forms germ layers, and the first embryonic tissues are formed. Some animals are diploblastic, meaning they produce embryos that only form two germ layers, the endoderm and the ectoderm, while some animals are triploblastic, meaning they form embryos that have three germ layers, the endoderm, the ectoderm, and the mesoderm. Most animals have tissues. What are tissues? We know that living organisms are made up of cells. Cells that are similar in structure and function will come together and work as a cohesive unit that perform specific tasks they are specialized in, and these units are known as tissues. Let's talk about the evolutionary history of animals. So it is believed that all animals share a common ancestor, and that common ancestor is also shared with a group of unicellular protists known as choanoflagellates that are thought to be the closest extant relatives to animals. It is believed that this common ancestor was a suspension feeder similar to choanoflagellates that we know. Let's talk about the history of animals through specific eras in time. Oldest, most widely accepted macroscopic fossils of belonging to soft-bodied eukaryotic animals that are collectively referred to as the Ediacaran biota, dated back to the Neoproterozoic era. The Paleozoic era. The Paleozoic era, like the other eras, is separated into smaller periods of time. One of these periods is known as the Cambrian period. And in the Cambrian period, a very significant evolutionary event occurred known as the Cambrian explosion. The Cambrian explosion was an explosive period of evolutionary change that gave way to organisms that more closely resemble the animals that we know, the animals of modern times. The Cambrian explosion also saw predators developing new adaptations to catch prey, like their claws, and prey animals developed new adaptations to protect them from predators, like their shells. And while these new predator-prey dynamics were on the rise, it is possible that natural selection resulted in a decline of the soft-bodied eukaryotes, soft-bodied animals like those found in the fossils that were dated back to the Neoproterozoic era, like the animals of the Ediacaran biota. And moving on to the Mesozoic era. In the Mesozoic era, Earth saw the first dinosaurs, it saw the first mammals, and it saw coral formations. Some species made the return from land back to aquatic habitats, and some species saw adaptations that enabled them to fly. The Mesozoic era, uh, it was concluded by a mass extinction event um, that wiped out the dinosaurs and gave rise to the next era, the Cenozoic era. The Cenozoic era saw a gradually cooling climate, and it also saw a rise in large mammalian species on the Earth. Let's talk about body plans of animals, some physiological characteristics. What is a body plan? A body plan is an archetypical set of characteristics that can be referenced when identifying a group of animals. One aspect of a body plan is body symmetry. Does the body have radial symmetry, meaning there's one axis on through the center of the body, and drawing a straight line division at any point crossing that axis will result in two mirroring halves. This is common in animals that live their lives attached to substrates of some sort. Then we have bilateral symmetry, which is the most common. It is the symmetry of our bodies. It is based around two axes, the head-tail axis and the ventral-dorsal axis. And lines can be drawn that will divide the body into mirroring right and left halves. Or some animals have no symmetry, like many sponges have irregular shapes and they lack any sort of symmetry at all. You can't really divide their bodies in any way and get mirroring halves. Most animals have tissues, like we discussed before, and animals that do have tissues are classified as eumetazoans, and they belong to a clade called Eumetazoa. Another important part of a body plan is, before we move on to talking about body cavities, I want to talk a little bit about the germ layers that are present in animal embryos that have undergone gastrulation. Starting with the ectoderm, the outermost germ layer. From the ectoderm, your skin, your outermost layer, and your central nervous system will be formed. Your endoderm, or the endoderm, is the innermost germ layer of an animal embryo that has undergone gastrulation, which surrounds the archenteron and lays the foundations of the digestive structures and some of the internal organs of the animal. Now, all animal embryos have the ectoderm and the endoderm, but only triploblastic animals form embryos that develop a mesoderm, a third germ layer. The mesoderm is the third germ layer, and it exists between the ectoderm and the endoderm. It's in the middle. It is present in all animals that exhibit bilateral symmetry. All animals that exhibit bilateral symmetry are classified as triploblastic animals because they form a mesoderm. Their embryos form a mesoderm. And the mesoderm is responsible for laying the foundations for your muscles and the internal organs that are not formed by the endoderm. So let's talk about body cavities now. Almost the vast majority of animals have a fluid-filled body cavity of some sort. Fluid meaning it could be a gas, it could be liquid. But a fluid-filled body cavity of some sort that provides protection, supports structural integrity, supports the internal organs, and facilitates resource transport within their bodies. Columns are cavities that are located within the mesoderm. They support a collimate's, which is an animal that forms a column. It supports a collimate's internal organs and are only present in triploblastic animals, because they're products of the mesoderm. A hemocole is another kind of body cavity that is exclusive to triploblastic animals, but they form between the mesoderm and the endoderm rather than within the mesoderm, and are filled with nutrient transport facilitating fluid known as hemolymph. Some animals do not have body cavities at all, like very flat worms, for example. Moving on from body cavities to development modes, some animals undergo protostome development, which is primarily differentiated from deuterostome development by discrepancies in cleavage and the formations of columns, and also whether the mouth or the anus forms first. Protostome development. Animals that exhibit protostome development, their their mouths form before their anus, and animals that exhibit deuterostome development are the opposite. Their anuses will form before their mouths. Animals that exhibit protostome development undergo spiral determinate cleavage, which means that cell division occurs on planes that are diagonally situated from the embryo's vertical axis. So it makes it look like a spiral. And also, because the cleavage is determinate, each embryonic cell cannot become a fully viable embryo on its own. Now, animals that exhibit deuterostome development undergo radial indeterminate cleavage. This means that cell division occurs on planes that are situated either perpendicular or parallel to the embryo's vertical axis, and each embryonic cell does not have a predetermined fate. So any individual cell has the potential to form a viable embryo on their own. In protostome development, the archenteron, as the archenteron is formed, masses of mesodermic tissue split apart and form the column. Additionally, the animal's mouths are formed from the first opening of the archenteron, which is the blastopore. And this harkens back to what I was saying about the differences in whether the mouth or the anus forms first from the blastopore. So protostome development, the mouth forms from the blastopore, and in deuterostome development, the anus forms from the blastopore. Oh. Another interesting thing to talk about when considering body plans of animals is the presence of a backbone. Some animals are invertebrates, which means that they do not have a backbone, and they make up the vast majority of animal species, with the only phylum that contains vertebrate animals being the phylum Chordata. Vertebrates are animals that do have a backbone. They are vastly outnumbered by invertebrate animal species. And finally, let's get into the phylogeny of animals. So, as you can tell from what we've talked about so far, animals are very diverse. They come in all shapes and sizes, very different structures, different body plans, different cavities, different types of symmetry. Even though they are all descended from a single common ancestor. Sponges actually belong to they are a basal taxon to all other animals. They diverged very early on in the evolutionary history of animals. Most animals are bilaterians and have tissues. Bilateral animals belong to the clade Bilateria, which is characterized by the presence of bilateral body symmetry. Bilaterians can be more specifically categorized into three main groups. Deuterostomia, which is one of the three main clades of bilaterian animals and is the only one of the three clades that includes vertebrates, because the phylum Chordata belongs to this clade. Another group that you can classify bilaterians in is Lophotrochozoa, which is one of the which is a major clade of invertebrate bilaterians, which is characterized and named for both the lophophore, which is a ring of tentacles that aids in feeding that is present in some species, and the developmental trochophore larva stage that is part of the life cycles of some species. And finally, we have the clade Ecdysozoa. It's the other of the major clades of invertebrate bilaterians, whose members are characterized by their common ancestry, but the clade is named for the ability to secrete exoskeletons that cover them and protect them, like such as some insects that will form an exoskeleton and then molt it whenever they grow too much, when they get too large for the exoskeleton, and then they will secrete another. In regards to the future of animal systematics, our understanding of animal systematics continues to evolve and is always subject to change with regard to new discoveries. Much of what we know is not set in stone and probably will change at some point. I've just listed some key terms here. I think I covered most of them. All of them. And thank you for listening. That's my presentation. Thank you so much.